Welcome back. So now our next concept is indicators. So we looked at the concept of pH value. So now we're going to go into our indicators. What are they and how do we use them? So we need to know that some substances have a different color in acid compared to a base. So there's a certain substance that has a different color if it's in an acid and also a different color if it's in a base. Now those substances, they are referred to as acid-base indicators. So if an indicator or an acid-base indicator, it is that substance that determines if whether a substance is acidic or basic by changing its color. So different indicators change color at different pH value. So we can have multiple indicators and they will each change color differently depending on the pH value. You might find that if you pour an indicator into a strong acid, the color change is going to be red. If it's into a weak acid, it's going to be orange. So you'll notice that the color changes between a strong acid and a weak acid are different. And that is because there is a difference in the pH value of the two substances, either a weak acid and a strong base, or a weak acid and a strong acid. Now, how do we then use indicators? We add an indicator to a substance to indicate whether a substance is acidic or basic by observing the color change. So the important thing is that we add the indicator into a substance that we want to test, and then the only thing that we need to do is to observe the color change. Remember, it is not advisable for us to taste. We just need to observe the color change of that particular reaction. Now, this table shows a selection of acid-base indicators and the color they will have at different pH values. So we can have, as we've said, we can have multiple indicators and they will each change color differently depending on the substance that you are testing, either if it's an acid or if it's neutral or if it's a base. For instance, we can take, for instance, the red onion water. So the red onion water can be used as an indicator. If it's poured, if few drops of it are poured into an acid, then there's going to be a color change to red, right? And then if the substance is neutral, so the color is going to be violet. It's going to remain violet. And then if the substance is a basic substance and you pour the red onion water, then there's going to be a color change from violet to green. So in the neutral substance, if the pH is 7, in a neutral substance, there won't be any color change. So therefore, we can also use a red litmus paper. We know that red litmus paper will remain red in an acid. And then if it's dipped into a base, it is going to turn from red to blue. So also again, if it is in a neutral substance, it will remain red as well because there won't be any reaction between the substance and the litmus paper. So we have different types of indicators. We have natural indicators and we also have man-made indicators. Now, what is the difference between the two? So natural indicators can be extracted from plants. For instance, we can use the juice of berries. Berries as an indicator. So we're going to take a few drops of it and pour it into either a substance being a base or an acid because we want to check if whether the substance is an acidic substance or a basic substance. So as we can see, we are pouring those drops of the juices of berries as an indicator into the substance being a base. Now you notice that there is a color change in that substance. It started as purple and then after pouring that indicator, it turns to gray. So because of that, we are able to tell if whether the substance is acidic or basic by observing the color change. Now, as I've said, we're just going to be adding drops of the indicator to test if the substance is acidic or basic by just merely observing the color change. There is no tasting involved. Now, we also have litmus indicators. Now, litmus paper does not change color in the presence of a neutral substance. So litmus paper as an indicator, so this is our indicator, if it's dipped into a neutral substance, it will not change color, right? However, it is going to respond to acids and bases in the following way. So if that litmus paper is dipped into an acid or dipped into a base, it is going to respond. However, if it's dipped into a neutral substance, there won't be any response. It will just remain that particular color. If it's red, it will remain red. Now, how does it respond? So if you dip a blue litmus paper into an acid, it is going to turn red. So blue litmus paper turns red in the presence of an acid. And red litmus paper remains red in an acid. So if it's a red litmus paper, it will not change if you're dipping it into an acid. But if you dip a blue litmus paper, as you can see, onto an acid, there's going to be the color change. The color change will only appear on the litmus paper itself, not the entire solution. So the litmus paper itself will turn red. Now, what about now if it's in a base? The red litmus paper will turn blue in the presence of a base. And if you're using a blue litmus paper and dipping it into a base, it will remain unchanged. It will remain blue in a base. But in an acid, the blue litmus paper will turn red. As we can see from the picture, we have our litmus paper, dipping it into an acid, then there's a color change that is being observed on this litmus paper. Notice that no one tasted anything here. You shouldn't too. So now, how do we do that? We dip the litmus paper into the substance to be tested so that we can observe the color change. I can't stress this enough. We do not taste anything. Now, also, we said that we have natural indicators and now the man-made indicators. So the difference between the two is that some indicators are large man-made molecules, like we have the bromothiamol blue as one of our indicators, and we also have the phenolphthalein as also one of our indicators. So these two, they are man-made indicators. And they're also dissolved in alcohol. The same thing happens. We just pour a few drops of either the bromothiamol blue or the phenolphthalein onto the substance that we want to check, that we want to test. And then we are going to observe the color change after having poured the few drops of either the bromothiamol blue or the phenolphthalein. Now, a special indicator that is so sensitive, it does not only tell us whether a substance is an acid or a base, however, it also tells us its approximate pH. So what is this particular substance? Now, this is also an indicator called a universal indicator. So unlike litmus, universal indicator can show us much more accurately how acidic or basic a solution is. So we know with the litmus paper, we're just going to observing the color change. We don't know how acidic it is. We just know that it's an acid or it is a base. With the universal indicator, it is going to tell us that this substance is acidic and how acidic it is in terms of the strength. How acidic, what is the pH in terms of the pH value? Where do you locate it on the pH scale? So that's how accurate the universal indicator is. So universal indicator can change into a range of colors depending on the pH of the solution. So remember, with the other ones, with either the litmus or the bromothiamol blue or the phenolphthalein, we just pour a few drops, it changes into a specific color, and then that we can just check on the pH scale. Whereas with the universal indicator, it can change into a range of colors depending on the pH of the solution. As you can see, the color change that you're going to be observing will be checked against this picture here to say if whether your substance is red. That means it's a very, very strong acid, because as you can see, you count, there's 14 test tubes here. The 7th one will be the neutral. And then as you move away from 7, remember the strength increases. Going towards 0, it becomes more acidic or more stronger. Going towards 14 on the right-hand side, it becomes more basic or more stronger of a base. Now, in the following picture, as I've said, solutions of increasing pH were mixed with universal indicator to show its full range of colors. So we have different substances that were mixed with the universal indicator to show the range of the universal indicator in terms of its colors. So, a universal indicator also, it is a combination of indicators and has different colors for weak and strong acids and strong bases. So we know that universal indicator has different colors for weak acids, different colors for strong acids, different colors for strong base, different colors for weak bases. Now, the color of the universal indicator gives the pH value of the substance tested. So once you pour or once you dip the universal indicator into the substance, that color, it is going to give the pH value of that particular substance. whereas the other ones will just tell you if it's acidic or basic by just changing color, whereas the universal indicator gives you the specific pH value. Now, a typical universal indicator shows a range of colors at different pH values and distinguishes them as either strong or weak acid or strong or weak base. So that's how a universal indicator works. So now, the table that I'm showing here, it is that it shows the color of indicators at different pH values or different pH conditions. We have indicators on the left-hand side from the red cabbage water all the way to the universal indicator. And we are seeing how they change color if they are mixed with either an acid or if it's in a neutral substance or mixed with a base. For instance, litmus paper, we know. If it's in an acid, it will be red. If it's going to be in a neutral substance, it will remain unchanged. But if it's in a base, it is going to turn blue, from red to blue. And then whereas this one will turn from blue to red. So we can see all the other indicators, how they change color depending on the substance that they are trying to test, either an acid or a base and even a neutral substance. So now, I hope now you understand a little better what the pH value is and also how to test if a substance is acidic or basic and even neutral. Now, with that said, I hope you had as much fun as I did. Until I see you next time, bye.